Welcome to today's biocast. Today we're going to be talking a little bit about mitosis. Mitosis is the process whereby we create new cells, and specifically mitosis is the creation and redistribution of the DNA so that we end up with two cells with the exact same complement of DNA in each of those cells. Now mitosis is done for every cell that you produce other than sperm and egg. When we create sperm and egg, we need to undergo a process called meiosis, or reduction and division. In meiosis, we need to end up with cells that have only one copy of each chromosome, or what are called haploid cells. In mitosis, we need to produce cells which are diploid, or have two copies of each chromosome. It can be confusing with meiosis and mitosis because they have the same names for the various stages they undergo: prophase, metaphase, anaphase, and telophase. Things to remember are, if you're making sperm and egg, you're doing meiosis. If you're doing anything else, making any other types of cells, you are undergoing mitosis. So what we see here is a single cell that is going to be starting prophase. Now, what we have in here we have our centrioles, these two can-like structures in our north pole and our south pole. This is going to be where we end up pulling our chromosomes towards. Now remember, your DNA is distributed in chromosomes. You get one set of chromosomes from your mom and one set of chromosomes from your dad. So each cell has two copies of each chromosome. So for example, we might have one chromosome that's produced and given to you from your dad in each cell, and one chromosome from your mom in each cell. Now, we're going to keep the shades similar so that the darker shade represents chromosome from dad, the lighter shade represents the chromosome from mom. So what we have here is one chromosome that has two copies. It's our blue chromosome. If we want to be able to create two cells that each have two copies of each chromosome, we have to duplicate each of these chromosomes. So we're going to duplicate them. So now we have one chromosome here and a second chromosome here. Now notice, even though there are now two parts, part 1, part 2, to each chromosome, we're still calling it a single chromosome. Each one of those parts, though, we call a sister chromatid. So this is a sister chromatid. This is the other sister chromatid of that chromosome from your father. Similarly, when we look at mom's chromosome, this is one sister chromatid. This is a second sister chromatid. So we have two sister chromatids comprising one chromosome. So we still only have two copies of our chromosome, 1 and 2. But each one of those chromosomes is made up of two sister chromatids. So if we look at what we have in humans, we have many more chromosomes than just the two. So we're going to throw a second chromosome in, and we're going to call this the red chromosome. This chromosome is from dad. This chromosome is from mom. And we're also going to throw in a green chromosome, this chromosome from dad, this chromosome from mom. So what we've had now in prophase is we've had the duplication of the DNA, and we've had that DNA condense out into chromosome form. Our next stage is going to be metaphase. In metaphase, these chromosomes are going to line up along what's called the metaphase plate, which is essentially just a region roughly dividing these cells. How they're going to do that is each one of these centrioles is going to send a microtubule from the centriole to our centromere, which I have represented as these round pieces in the middle of each chromosome and in the middle of each sister chromatid. This chromosome, the blue chromosome from dad, is going to get pulled towards the metaphase plate so that all of our chromosomes will eventually end up being distributed along that metaphase plate. So what we'll see is something that looks like this. remember, what we really have at this point is coming out of these centrioles, we have microtubules attaching to each centromere of the chromosome. And they're lined up along this rough metaphase plate. So we have these guys coming out here. And that's the end of metaphase. We've just basically lined our chromosomes up along the middle. Notice we still have 2 copies of the blue chromosome, 2 copies of the green chromosome, and 2 copies of the red chromosome. So our next stage is going to be anaphase. Now, anaphase, we have everybody lined up along the metaphase plate. We have microtubules running from the centriole to each centromere. We still have 2 copies of each chromosome, 1, 2, one of them coming from Dad, and the other one coming from Mom. We have 2 copies of our green chromosome, and we have 2 copies of our red chromosome. Notice the chromosomes don't need to be lined up next to each other, so the 2 copies don't have to be side by side like we see in the blue. In anaphase, what we're going to see happen is the microtubules are going to start pulling the chromosomes towards the 2 poles. The microtubule attached to this centromere is going to pull it up towards the northern pole. The microtubule connected to this centromere is going to pull it towards the south pole. So those tensions are going to end up breaking apart our 2 sister chromatids. So let's take a look. Okay, what we've seen is we have split apart that chromosome that was comprised of 2 sister chromatids, so that now we actually have 4 copies, 1, 2, 3, and 4 copies of each one of our chromosomes. Now actually in a tetraploid state. Now what we need to do is we're going to start separating these 2 cells. What we need to do is create a division so that we isolate this set of chromosomes from this set of chromosomes. Because as you can see, if we isolate it up here, we have 1, 2 copies of the blue chromosome, 1, 2 copies of the green chromosome, and 1, 2 copies of the red chromosome, and the same thing down here. So by isolating this set of chromosomes from this set of chromosomes, restore the diploid state to each of these 2 cells. And the process of that division is called telophase. We have 2 different ways that we can experience telophase. We can have the plant style, which results in cell walls being formed through the middle of the cell, so that eventually they link up and produce one cell in the northern pole and 1 cell in the southern pole. So now we have isolated these 2, and 2 cells will function independently. In contrast, when we look at animal cells, what we see is another microtubule that ends up spanning the cell and slowly constricting, like you would see in a balloon, so that we see a gradual pinching in as the microtubule that surrounds the cell collapses until it eventually pinches off and produces 2 unique cells. And that's the end of telophase. So this is the process of mitosis. We started out with 1 cell with 2 copies of each chromosome. We duplicated our chromosomes. We pulled those apart to the opposite poles. We created either a new cell wall or a plasma membrane between them. which resulted in 2 cells, each of which is diploid. So I hope that helps you understand mitosis, and we will talk to you at our next biocast.